Good evening everyone. Myself Noor from group 303 and today my topic is transfer mRNA and translation. First is role of tRNA in protein synthesis. Transfer RNA is an essential molecule in process of translation which is synthesis of proteins in cells. The primary role of tRNA is to transfer amino acids to ribosomes where protein synthesis occurs. So here we will first study the structure of tRNA. tRNA has a characteristic clover leaf-like structure which consists of three parts: anticodon loop, a three nucleotide sequence that pairs with complementary codon or messenger RNA or mRNA, amino acid attachment site located at 3' end where specific amino acid is covalently bonded. D loop and T loop. These loops help tRNA to fold into its correct shape and interact with the ribosome by amino acyl tRNA synthetase enzyme. Each tRNA is specific to amino acid and has unique anticodon that pairs with specific RNA codon. Next is process of translation. Translation occurs in basically three main stages. First is initiation, second is elongation, and third is termination. In this all three stages, elongation is the main characteristic stage or most important stage. Translation takes place in cytoplasm of eukaryotic and prokaryotic cells on ribosomes. First step is initiation. mRNA binding. The small ribosome subunits bind to mRNA molecules at start codon, that is AUG. Then tRNA binding. The initiation tRNA charged with methionine recognizes start codon through its anticodon, that is UAG, and binds to P site of ribosomes. Assembly of ribosomes. The large ribosomal subunits bind to small subunit and make a complex, forming the complex ribosome or complete ribosome ready for elongation. Second step, which is the most characteristic step and important step, is elongation. Codon recognition. A charged tRNA enters a site of ribosomes, pairing its anticodon with the mRNA codon. Peptide bond formation. The amino acid in P site forms a peptide bond with amino acid on A site. This is catalyzed by enzyme peptide transferase activity in ribosomes. Last step is translocation. The ribosome moves one codon down, the mRNA shifting tRNA A to P site and P to E site, means exit site, where it is released. Last step, this process repeats as polypeptide chain grows, and the last step is termination, which is synthesized by stop codon recognition. When a stop codon UAA, UAG or UGA enters A site, no tRNA can bind to it. Instead, these are release factors promoting release of polypeptide chain. Dislocation of ribosomes. The ribosomal subunits disassemble and mRNA and tRNA are released. Now, before the process of translation, amino acylation or tRNA charging is must for the corresponding amino acid to be joined to it. What is amino acylation or tRNA charging? Before translation, each tRNA must be charged with its corresponding amino acid. This is done by an enzyme called as amino acyl tRNA synthetase. The enzyme catalyzes the attachment of amino acid tRNA, creating an amino acyl tRNA complex. This ensures that RNA carries the correct amino acid corresponding to codon it recognizes. Thank you.